Hello guys, welcome to my channel. In the previous video we studied about the folding patterns of the carbohydrate and also we studied about the other polysaccharides, right? And agar and agarose. And so we'll continue with the polysaccharide. And now, so we studied the cell wall of the polysaccharide, right? The homosaccharide that covers the cell wall. But now we'll study about the extracellular matrix. So as we know, the extracellular space in the tissues of multicellular animals, right? So there is a cell and there is an extracellular space outside the cell, right? So the extracellular space in the tissues of multicellular animal is filled with the gel-like material that is called extracellular matrix, right? As we all know, extracellular matrix. So that is called extracellular matrix or ECM in short, right? So this extracellular matrix, also called a ground substance, which hold the cells together. Like there are different bunch of cells and there are like extracellular matrix outside the cell. So what the extracellular matrix do, it holds the cells together and provides a porous pathway for the diffusion of nutrients and oxygen to individual cells. So from one cell to another cell when it goes, nutrients or oxygen. So this provides a porous pathway. So the ECM, the extracellular matrix, also surround the fibroblast and other connective tissue cells that is composed of an interlocking meshwork of heteropolysaccharide. Right. So this extracellular matrix that also surrounds the fibroblast and other connective tissue cells as well. So this is composed of interlocking meshwork of heteropolysaccharide. As we are studying out the polysaccharide, so this is composed of heteropolysaccharide. Polysaccharides. So this is composed of heteropolysaccharide, and there are fibrous proteins with the heteropolysaccharide, such as fibrillar collagens, elastins, fibronectins. Right. So with this, do not forget there is a fibrous protein as well involved in the fibrous protein. Right. So for example, fibrillar there is fibrillar collagen, right? Then elastins or fibronectins and pronectins. So these are the example of fibrous protein, right? So this extracellular matrix is composed of the heteropolysaccharide and fibrous protein, right? So the basement membrane is a specialized ECM that underlies the epithelial cell. So when we study about the epithelial cells and the basement membrane, they're found, but just remember right now that basement membrane, what's called basement membrane? So the membrane that is specialized, basement membrane is a specialized ECM, the specialized ECM that lies underneath of epithelial cell. So underneath the epithelial cells, right? The basement membrane of that is specialized ECM. That ECM is consist of a specialized collagens, laminin, and heteropolysaccharide. So this ECM consists of specialized collagen, laminins, collagen, lamin, and heteropolysaccharide. So this is consist of heteropolysaccharide. And these heteropolysaccharides, which are this heteropolysaccharide that are referred, so that is glycosaminoglycan. So that we are going to study. So this heteropolysaccharide is glycosaminoglycans. So the glycosaminoglycans are a family of linear polymer composed of a repeating disaccharide units. Right, so the heteropolysaccharide, that is the glycosaminoglycans, what are they? They are a family of linear polymers composed of repeating disaccharide units. So what does that mean? So you can see a picture on your screen. I'm not drawing that long repeating disaccharide. So just you can see the image. So I will explain the image to you now. So as you can see, we are mentioning that it is a repeating disaccharide. So you can see the first one, hyaluronidase, and then the chondroitin sulfate, and it goes on and on as a repeating disaccharide, correct? And the molecules, these molecules are copolymers of alternating uronic acid and the amino sugars residues. So the first one is the uronic acid and the second is the amino sugar, and the chondroitin sulfate that you are seeing at a third, so that's an exception. So sometime it might occur, and with the sulfate ester in any of the several position except in hyaluronidase, right? And the ionized carboxylate and the sulfate group, so the one that you are seeing in the red, gives this polymer their characteristic high negative charge, right? So the combination of sulfate groups and the carboxylate groups, so you can see that both are negative, that is in red, of the uronic acid residues. This gives this aminoglycans a very high density of negative charge, and to minimize the repulsive force among the neighboring charge group, right? They are both negative, so when negative cannot go together, so to minimize that repulsive force among these neighboring charge groups, these molecules assume an extended conformation in solution, forming a rod-like helix in which the negatively charged carboxylate groups occurs on the alternate side of the helix, right? This is how it shows in heparin, the one that you can see in the heparin, the red, green and blue molecules. So you can see the oxygen and sulfur are in the red color, red and yellow, right? And the carbons in the hydrochloric acid sulfate are blue, but those in the glucosamine sulfate are green, right? So this is how it opposes try to stay away from each other, so they minimize the repulsive force. These glycosaminoglycans are unique to animals and bacteria, but these are not found in plants. And one of the two monosaccharide is always either an N-acetyl glucosamine or N-acetyl galactosamine. So one of the two monosaccharide in this heteropolysaccharide will be N-acetyl glucosamine or N-acetyl. One of the two monosaccharide will be N-acetyl glucosamine and N-acetyl galactosamine. So this in this heteropolysaccharide, so one of the two monosaccharide will be N-acetyl glucosamine, or it can be as N-acetyl galactosamine, and the other is in most cases a uronic acid, and the other in most cases is uronic acid, right? That you can see in the hyaluronidase. So this uronic acid usually will be D-glucuronic acid or L-hydruronic acid. So this can be either D-glucuronic acid or L-hydruronic acid, the uronic acid that is present. So some aminoglycans contain esterified sulfate groups as well. Alright, so the specific patterns of the sulfated and the non-sulfated sugar residues. So like in the picture, the one with the SO3, that is a sulfated group, right? The chondroitin sulfate or the keratin sulfate, and the one hyaluron has a non-sulfate sugar residue. So this, what's the difference between that? So that allows glycosamine glycans allows specific recognition by a variety of protein ligands, so that binds electrostatically to these molecules, right? So the sulfated glycosaminoglycans are attached to the extracellular proteins to form proteoglycans, right? So the sulfated glycosamine glycans, what are they do the what they do so they are attached to the extracellular proteins to form proteoglycans, right? So the aminoglycans, hyaluronon, so the hyaluronic acid contains alternating residues of D-glucuronic acid and the N-acetyl glucosamine. So the first one in the picture, the glycosaminoglycan hyaluronon, so what it has is a repeating, so it has a D-glucuronic acid and an acetyl glucosamine as an alternating structure, and with up to 50,000 repeats it can have of the basic disaccharide unit, right? And a hyaluronan has a molecular weight of several millions, right? And it forms a clear, highly viscous, non-compressible solution that serves as a lubricant in the synovial fluid of joints and give the vitreous humors of the vertebrate eye its jelly-like consistence, right? So we'll write the uses of the glycosamine, aminoglycan. So what are the uses? So this hyaluronan is clear, right? This is clear. It is highly viscous. It is non-compressible, right? So these are the properties. It is non-compressible solution. So this is a solution, and what it does, it that the function that serves as a lubricants in the where it serves as a lubricant in the synovial joints, right? Synovial fluids. It serves in the synovial fluids of the joints. And also what it does, and give the vitreous humor that is in the eye. Humor, it's jelly like. So this is vertebrate, all vertebrate eye, right? Vitreous humor, it's in the vertebrate's eye, and that gives its jelly-like structure, and by its name hyaluronan. So in Greek, hyalos means glass, so that's why it gives like a glassy or translucent appearance. So that was the one function. So there is another function for this hyaluronan, is this is also a component of extracellular matrix of cartilage and tendons as well. So it is also a component of cartilage and tendons, right? That is ECM, right? I'm not writing in full, but it's a component of extracellular matrix in the cartilage and tendons. So what it does, it contributes tensile strength and elasticity as a result of its non-covalent interaction with other components of the matrix. So what it does, it provides strength and elasticity, right? How? Why it provides, how it provides, as a result of its strong non-covalent interaction with other components of the matrix. So these are the two functions of the hyaluron glucosaminoglycan hyaluron. So there is an enzyme hyaluronidase, hyaluronidase, right? So there is an enzyme. So this enzyme what is secreted by some of the pathogenic bacteria, right? So for this there is an enzyme hyaluronidase. It's secreted by some of the bacteria, pathogenic bacteria. What it does, it can hydrolyze the glycosidic linkages of this hyaluronidase, rendering the tissues more susceptible to the bacterial invasion. So how the bacteria are invaded in the tissues? So some of the bacteria have this enzyme and they secrete the enzyme, and that enzyme, and I will write the pathogenic enzyme, pathogenic bacteria. Enzyme, right? So the enzyme will break down the bonds of this hyaluronidase, that is the glycosidic bond, and then the bacteria can easily invade the tissues, right? And in many animal species, the similar enzyme is in the sperm hydrolysis as well. So the outer glycosaminoglycan coat around an ovum, allowing sperm penetration. This glycosaminoglycans hyaluronidase, there are other glycosaminoglycans that differ from this in three respects. So they are generally much shorter polymer, and this is like really long. It has 50,000 repeats, right? So the others are very much shorter. They are covalently linked to a specific protein, that is proteoglycans, and one or both monomeric units differ from those of hyaluronidase. So like the monomeric unit, it is like this and this. So the other are different from the monomeric unit. It might have a different kind of monomeric unit, right? And it is covalently linked specific to proteins, right? Specific protein. So the other is chondroitin sulfate, as you saw in the picture, right? The second one, chondroitin sulfate. I misspelled it. This chondroitin sulfate, right? So this is in Greek. It's chondros means cartilage. So that's why, if you remember, chondroitin sulfate contributes to the tensile strength of the cartilages, tendons, ligaments, heart valves, and walls of aorta. So the functions of this chondroitin sulfate, what it's done, it controls tensile strength of cartilage, right? Tendons, it also can ligaments, right? Heart valves, the walls of aorta. So this is all have this chondroitin sulfate heteropolysaccharide. There is other as well. This is first, this is second. Right, first, this is second, and there is third one as well. So that is dermatan sulfate. Okay, write it down here. Dermatan sulfate, right? So this contributes to the pliability of the skin and is also present in the blood vessels and heart valves. So the dermatan sulfate, what it does, it contributes to the pliability of the skin. Right, pliability of skin, and is also present in blood vessels and heart valve too. All right. So in these polymers, many of the glucuronate residues present in the chondroitin sulfates are replaced by C5-epimer and L-iduronate. There are two more this. I will see if this base is one. Right. So there is dermatin sulfate, chondroitin sulfate, and the fourth one is keratin sulfate. Keratin sulfate. So the keratin, that is keratin, right? Keras means horn in Greek. So this have no uronic acid. Does not have no uronic acid. You know what is the uronic acid means? Like that contains carboxylic and carbonyl group as well. So when you see the structure, so the image that you are seeing on your screen, that's for the keratin sulfate. It does not have the uronic acid that are present in this hyaluron, chondroitin, or like dermatin. So keratin sulfate, no uronic acid, and their sulfate content is variable. So where this keratin is present, they are present in cornea, cartilage, bone, and a variety of horny structures as well. Has a name in keratin. So this are present. This is present in cartilage, right? Bones, and other horny structure. What kind of horny structure it is? Like horns, hair, nails, right? So this horny structure are formed from the dead cell and like horn, hair, nails, and claws, right? So this is keratin sulfate, and the fifth is heparin sulfate. Alright, so this is the last one, heparin sulfate. So heper means liver. So this is actually originally isolated from the dog liver first, and this is produced by all animal cells. This is produced by all animal cells and contains variable arrangement of sulfate and non-sulfated groups. So the sulfated segments of the chain, right? The sulfated segment of the chain of the heparin allowed it to interact with a large number of proteins, including growth factors and ECM component, as well as various enzymes and factors present in the plasma. So heparin is highly sulfated intracellular form of heparin sulfate produced primarily by mast cell. What is a mast cell? That is a type of leukocyte or immune cell. So this heparin sulfate has sulfated and non-sulfated group, right? Sulfated and non-sulfated, right? So the sulfated segment of the chain, that is this one, the sulfated segment of the chain, what it does, it allows it to interact with the large number of proteins. So this allows, sulfated segment allows to interact with large number of, right? It allows to interact with large number of proteins. That includes growth factor and ECM, that is extracellular matrix components, right? Not extracellular matrix, but it's components, and also enzymes and factors present in the plasma. Enzymes and factors present in plasma. So the heparin is highly sulfated. This heparin is a highly sulfated intracellular form of heparin sulfate produced primarily by mast cell. So this is highly sulfated, and the heparin sulfate is actually intracellular form of heparin sulfate. So the intracellular form of heparin sulfate is produced by actually mast cells. Produced by mast cells. And what are the mast cells? So mast cells are a type of leukocyte, or you can say an immune cell as well. But the physiological role of this heparin sulfate is not yet clear. But as we all know, the purified heparin is used as a therapeutic agent to inhibit coagulation of blood through its capacity to bind the protease inhibitor antithrombin. So why this is used as a and inhibit coagulation of blood? Therapeutic. Use as a therapeutic agent. Why it uses this as a therapeutic agent? This is to inhibit coagulation of blood. How it does? Through its capacity to bind the protease inhibitor antithrombin. So that is why this is used as a therapeutic agent. All right, that was the 5 glycosaminoglycans: hyaluronidase, chondroitin sulfate, dermatan sulfate, keratin sulfate, and heparin sulfate heteropolysaccharides. Right, guys, so that was for the polysaccharide. I think that this is really enough for this video. All right, thank you guys for watching. All right, we'll see you in the next video. Thank you guys for watching, and don't forget to subscribe.